A very lovely good afternoon to all of you. I am Dr. Pooja, and I've been again discussing the biochemistry portion as well as the questions with you since like 7 8 months. So after a long time, after really a gap of 15 20 days, I will be recording again a video for you guys. Since the exams are nearby, one of the exam is going to be conducted in the month of December, and so to prepare you for that exam as well as the exam which is going to be conducted next year. So today my topic of discussion would be the questions based on the metabolism of proteins. So let's start up with the discussion. Now, all of you know proteins, if we talk about these are what? These are the polymers of the amino acids. So whatever is the property of the protein, that is actually coming from the amino acids which are making it. So whenever we talk about the protein metabolism, we have to actually deal with what? The amino acids. And always remember these amino acids have a zwitterion structure. Now, what do you mean by this zwitterion? Zwitterion means they can behave as acid as well as bases, or you can say they are the amphoteric substances. In a sense that they can be acids, they can donate a proton, they can be bases, they can accept a proton, and that is why they are amphoteric. And in general, if we see the structure of a peptide, amino acid, you can say it is like this. This is the structure, generalized structure, and this R is the one on which the entire classification of the amino acids is based. When one amino acid combines with another amino acid, second amino acid, it loses a molecule of water, forming a peptide bond. Keep on adding the number of amino acids and we get a polypeptide. So if we have to talk about the metabolism of proteins, we have to study the amino acids. Now let's start discussing the questions. A patient diagnosed with Hartnup's disease due to defective transporter required for the absorption of amino acid tryptophan has been brought with skin rashes and suicidal tendencies. Now, tryptophan is a precursor for so many compounds, the deficiency of which can cause the said symptoms, and which out of the following is not synthesized by tryptophan. Now see, Hartnup's disease, always remember, it is actually due to defective reabsorption of neutral amino acids or tryptophan. Now the patient, he is diagnosed with what? Obviously Hartnup's disease, and has been brought with skin rashes and suicidal tendencies. And we know tryptophan is required. It is the precursor of serotonin and niacin in our body. 60 milligram of tryptophan is required to synthesize 1 milligram of niacin. So these are the two very important products, and serotonin, which is further converted into melatonin. So these three are the very important products which are synthesized by essential amino acid tryptophan. But the question says skin rashes and suicidal tendencies. Fine, we can say serotonin is what? It is a potent vasoconstrictor. It stimulates the cerebral activity, right? And once it stimulates the cerebral activity, once its function is over, it is degraded to form 5-hydroxyindole acetic acid. But here the question is simple. Nothing you have to discuss. You just have to say which out of the following compounds is not synthesized by tryptophan. So just now I mentioned serotonin, melatonin, and niacin. Only epinephrine is not synthesized by tryptophan. And the precursor of epinephrine is tyrosine. Very simple question. So it would be this. Second question, the synthesis of all of the following compounds except this deficient in a patient suffering from phenylketonuria. Again, an indirect question, I should say. Phenylketonuria is due to the deficiency of phenylalanine hydroxylase. This enzyme converts what? Phenylalanine to tyrosine. Right. So phenylalanine is converted into this, and if this enzyme is deficient, the subject will suffer from phenylketonuria. Now the question is the synthesis of all of the following except deficient in a patient suffering from this. Now you know tyrosine is a precursor of T3 and T4. Tyrosine is a precursor of melanin, and it is a precursor of catecholamines in our body. And if we look at the options, you can see melanin is synthesized, yes. Melatonin is not synthesized. It is synthesized from tryptophan. But these things you have to learn. These are very, very important. And catecholamines are epinephrine and norepinephrine. And thyroid hormones, these are all synthesized by tyrosine. So this person suffering from phenylketonuria will be deficient of all the metabolites except melatonin. So the answer to this question is melatonin or B. Next question, histamine, a chemical mediator of allergies and anaphylaxis synthesized from amino acid histidine by which of the following process? Now histidine, a very important basic amino acid and the most stable amino acid at physiological pH. Right. If it undergoes decarboxylation, means if it undergoes removal of carbon dioxide, what is left? Only the amino group. So histidine via decarboxylation will be converted into histamine. Histamine, which is actually released during allergies. So obviously the process is a removal of carbon dioxide. It is decarboxylation. So the answer would be B to this. If you look at other processes, deamination, D means removal of amino group would be deamination. Transfer of an amino group will be transamination. Removal of hydrogen will be dehydrogenation. But here the question is simple, histamine, amine. And basically it is clear from the word itself. If amine is there, carboxylation or you can say carbon dioxide is given out. So it would be histamine is synthesized by decarboxylation. Next question number 4. The diet of a child suffering from MSUD, maple syrup urine disease. It is actually due to the deficiency of alpha keto acid dehydrogenase enzyme. Right, and this is actually due to the branched chain amino acids, BCAA, and the mnemonic for this is LIV, leucine, isoleucine, and valine. So these three amino acids are actually the branch chain amino acids whose metabolism is like somewhat similar. Deficiency of this will lead to maple syrup urine disease. The urine gives the smell of burnt sugar. Urine gives the smell of burnt sugar. Right. And out of these, if we say leucine is considered to be a potent ketogenic amino acid. Right? So the answer is A. Let's move on to the next question. Which out of the following amino acids is not required in creatine synthesis? Now see, this creatine is what? It is the energy reservoir of the muscles, right? And it is synthesized by three amino acids. It says it is actually a tripeptide. which is synthesized from 3 amino acids, G, A, M. G stands for glycine, A stands for arginine, and M stands for methionine. Right? And this reaction takes place in liver as well as kidney. In the liver, transmethylation occurs, which is by methionine, and in the kidney, glycine and arginine combine to form guanidoacetate. Now let's look at the question, which out of the following amino acids is not required in the synthesis of creatine? GAM. So B. Serine is not required for creatine biosynthesis. And creatine, when it gives out a molecule of water, it gets converted into creatinine. Right? Then urea is synthesized in. Now see, urea is synthesized in what? There are 2 organelles required for the synthesis of urea. And I'll tell you one mnemonic also. There is a mnemonic called HUG. HUG in H stands for heme synthesis. U stands for urea cycle. And G stands for gluconeogenesis. So you can say these 3 processes takes place in 2 organelles. And that is the question. That is what the question says, that urea is synthesized in. So urea is synthesized in both cytoplasm as well as mitochondria. And this urea cycle consists of 5 steps. First and second takes place in the mitochondria, and third, fourth, and fifth takes place in the cytosol. Right? Then next similar question again from urea cycle. The 2 nitrogen of urea are derived from. So urea cycle has this formula. Urea has this formula, NH2CO and H2. Here this C comes from carbon dioxide, which is present in the form of bicarbonate ions. 1 nitrogen comes from obviously the amino acids, because amino acids are the one which are expelling this toxic ammonia in the form of urea. And the other nitrogen will come from aspartate, aspartic acid. So let's look at the options. The 2 hydrogen are derived from aspartate and ammonia. So the answer would be A. I hope this question is again clear to you. These are very simple questions. Now the first line of defense in brain in conditions of hyperammonemia is, you know, ammonia is highly toxic in action, means it cannot move in a free form. So it requires a transporter which will carry this toxic ammonia. So that transporter is what? It is glutamine. Right? And synthesized from glutamate. So glutamate, when it combines with toxic ammonia, it forms this glutamine. By the enzyme glutamine synthetase, and this glutamine handovers this toxic ammonia to the liver, where it undergoes urea formation. Right? So the first line of defense in brain in conditions of hyperammonemia is we have to increase the synthesis of glutamine. And that is why we say N-acetyl glutamate also increases the formation of urea. And NAG is a positive modulator of urea cycle, or it forms carbamoyl phosphate. Right? Then which out of the following is required as a coenzyme for transamination? Very important question. Always remember, transamination requires PLP or vitamin B6 or pyridoxine. The question can be asked in either of the ways. No transamination is possible without pyridoxal. Coenzyme A, if we say coenzyme is pentotheniine or pantothenic acid or B5. Folic acid gives rise to 1-carbon metabolism. Right. So coenzyme for transamination is pyridoxal phosphate. Then in a patient suffering from cystinuria, very important, again it is a neutral diabasic amino acid. Which of the following is not seen in the urine of affected patients? So if there is defective tubular reabsorption, or you can say defective reabsorption, what will happen? There will be excretion of 4 amino acids in urine in a person suffering from cystinuria, and they will be COLA, C-O-L-A. Cysteine, ornithine, lysine, and arginine. So if a person is suffering from cystinuria, we can expect excretion of these 4 amino acids because the protein which is transporting them, it is defective. So it is COLA, you can see. Only methionine is the one which is not seen in the person suffering from cystinuria. So today we discussed just 10 questions. This was a very small session. Just to, start up with this so that at least you can have an idea of what kind of questions can be expected in metabolism of proteins. So I hope I was able to satisfy some of your queries, and these are the questions which are important for the students who are preparing for this MSc also. MSc entrance for biochemistry. Right? So this is all for now. If you guys have any problem, you can log into my website biochemistrysimplified.com. And if you like my channel, subscribe to my channel, give maximum likes to me, and you can also press the bell icon so that you can get the videos in future also. So wish you guys all the best. Take care. Have a good day.